yesterday, you know how our game is played. Remember, let's try to keep on questions that are focused on the topics that were just presented by the panel. And if it's something very, very specific to your situation, your meeting request, the panel's probably not going to be able to respond to that. So you might want to submit those more properly to the agency. Again, if you've got questions here in the room, we've got 2 microphones. Please line up in front of those microphones. And online, if you have not already submitted your questions, you can use that Q&A pod that's in the lower right-hand corner. Type your questions in. If it's for a specific person, please type their name in ahead of time. Let's go ahead and start at microphone number 1, please.
Hi. First off, I'd like to thank all the speakers for nice, clear, concise presentations. I really appreciated that. This question was first brought up or came to mind with Dr. Ghosh's presentation, but I've heard it a lot throughout the conference. And could you give me a definition or at least some examples of what you mean when you say inherent or obvious bioavailability or bioequivalent, in your area?
Can you repeat the question, please? I didn't understand the last part.
At the beginning of your talk, you mentioned when you were giving criteria for what you need to look at for bioavailability. Often I hear the very first thing that it's obvious, it's inherent. You might have used a different term. I apologize for my
bioavailability is self-evident.
Mm-hmm.
So what we mean by that is that there is no microstructure or any other arrangement of matter in the formulation itself that could impact the release of the API from the formulation. So if it's, say, for example, a simple solution where there is no structure that would prevent or that would impact the diffusion of the API in the formulation or the partitioning of the API from the formulation, that's what we mean by bioavailability may be self-evident because the formulation or the microstructure is not expected to impact the release from the formulation. And just to add one more thing, assuming that there are no changes in inactive ingredients, of course.
Okay, let's take our first online question, please.
Okay, this was a question that was presented to us during Dr. Ghosh's presentation. Dr. Rainey presented on the importance of Q1, Q2 to product bioequivalence. However, as Q1, Q2 for topical products is not required per the regulations, FDA is not obligated to respond to a controlled correspondence for Q1, Q2 sameness assessment per the most recent guidance. There therefore seems to be a disconnect between the guidance and the regulatory science. Can you please comment on whether controlled correspondence for Q1, Q2 assessment for topicals will be answered?
So if a product-specific guidance is available and it recommends a pathway for establishing BE that requires a product to be or recommends a product to be Q1, Q2 the same, then yes, that question can be answered using a controlled correspondence. However, if a product-specific guidance is not available at this time, then we recommend that if you are considering designing a formulation that is Q1, Q2 the same, and potentially choosing maybe an in vitro approach for establishing BE, you can provide us with the whole your whole product development strategy within the scope of a product development meeting package, and we should be able to assess the viability of that approach for the specific product in question.
Okay, and if there's any questions in the room, please come on up to the microphone. Oh, perfect timing. Nice and close to the microphone, please.
Yeah, thanks for the nice presentations. My question is to Dr. Ghosh. The Q1, Q2 sameness for topical products when there is a product-specific guidance is available. So we are asked to submit a control correspondence for assessing the Q1, Q2 sameness with RLD. But at times, RLDs are discontinued long ago. We don't find them anymore on the market. Then what is the recommendation in such cases?
So you are imagining a situation where the RLD will not be available for comparative assessment at all?
It's not an imagination, it's a fact.
I know, I know. My question meant that we won't be able to conduct, like it's not available, so comparative assessment of Q3 characterization as such, not only the deformulation is not feasible at all?
Yeah, that's correct. Q1, Q2, unless it's Q1, Q2, you don't get a bioever.
I think I'm going to let Sam handle this question.
So I appreciate, I think the circumstance that you're referring to is when the reference product is now maybe the reference standard, it's not the original RLD. Is that the kind of situation?
That's correct, yes.
So I think this is, from a regulatory perspective, it's a tricky question. It depends a little bit upon the particular drug product and what the regulatory history is for that product. What I specifically mean is what was the evidence that that established the current reference standard as the reference product. Depending upon the type of evidence that existed, it may be acceptable to use the current reference standard as a reference for the Q1, Q2, Q3 type deformulation and characterization. But that is a tricky one, and so that's a perfect kind of situation to be able to discuss in a pre-ANDA meeting request.
Oh, sure. Thank you.
Yeah.
All right. Thank you. Microphone number 1, nice and close for me, please.
This was just a little bit clarification for Dr. Sam. When we say that there is a plus or minus 5% variability that we can have with the RLD, does it mean like for each component, or is it like in general for it's like for the total formulation?
So that's a good question because it can be confusing. So when we refer to the plus or minus 5%, we're talking about each individual inactive ingredient. So for example, if a formulation contained 10% of ingredient X, then you could be between 9.5 and 10.5. So it's plus or minus 5% of that 10% on that inactive ingredient. And then likewise on another inactive ingredient. But what can end up happening is those differences can add up, and then what will end up happening is that, for example, the amount of water will no longer be within plus or minus 5% because all of your other little 5% have chewed away at it. So the whole formulation does need to essentially match where the inactive ingredients individually are within plus or minus 5% of that nominal concentration or the nominal amount.
So is there a range like how much the variability could be, like cognitively or additively?
I'm not sure I understand the question, but no, it's a specific plus or minus 5%. There's a little bit of allowable rounding on that. So you don't have to be out to multiple decimal places on that. But it's plus or minus 5% on each inactive ingredient.
That was reassuring. Thank you.
All right, let's take an online question, please.
Okay, this question is for Dr. Sakaloju. Can PK be used for establishing bioequivalence for non-Q1, Q2 formulation?
This is a good question, and it is a challenging one. And I think that our understanding at the agency is that the modeling and simulation tools to predict performance, in vivo performance for non-Q1, Q2 products are not mature right now. Of course, if an applicant would like to go that route, we'll be happy to receive the application and review, provide feedback, work together. But this is our understanding today. There are a lot of unknowns, and it is very important to make sure that the model captures those those formulation parameters and how they can be different between the RLD and the test product. So this is definitely our ultimate goal, but we don't think that we are there yet. Yes.
Sam, want to add anything?
Yeah, I want to make sure that it comes across encouraging in response to that question, that modeling and simulation may in fact have a very good application to situations where it's not exactly Q1, Q2, that there are small differences. And the tricky part that I think leads to some tentativeness that Eleftheria is referring to is that it's challenging to verify the model. Where will you get the underlying data to verify the model so that the model can then be utilized in that circumstance? So I think the answer is that would be an ideal situation in which to use modeling. The challenge, and we would be happy to work with you on this, is how do you verify the model? Would you add anything to that? Okay.
All right, microphone number 2, nice and close.
Hi, I'm Maria Flynn from the OPQ. First, I want to congratulate my colleagues. You did a wonderful job at being very explicit and filled with a lot of very valuable information. I just made some little bit of notes in the means of being very generous to our industrial partners here of some things that I thought I'm still seeing a lot of, even though there's guidances and also USP that will help people in the topical world. One thing, USP 3 goes into clear information about globular size and also the lack of crystallinity in the case of something that's all uniform. We're still seeing a lot of deficiencies with that, so please take note of that. Also, container closure. There's a lot of problems sometimes with the semi-permeable. Look at stability guidance Q1A for that in terms of % weight loss. We're still seeing situations where people are not giving us that data for the validation batches where it seems risk-based needed. Also, with container closure, when there's aluminum and there's a laminate, the container integrity of the liner is still being cited a lot for deficiencies there. So please include a task for that container integrity. As well as in the cases where it's not pump or meter dose for device, but where there's functionality important, such as a pump, that also is very important to do some kind of minimal basement requirements, such as like priming the pump and so on. Some of these things are less obvious sometimes and not very product specific. And then one other little comment in terms of benzoquinone. That seems to be a major deficiency that we're seeing in cases where it's applicable, of course, where there's semi-permeable containers being used. As many of you know, in 2014, it was named for topicals as a very allergic reaction. So we like to see the leachable, extractable data for that. And again, this is all my own opinions, but these are common things that I've been seeing. They haven't been really emphasized. And I just wanted to give some generous tips.
Thank you for those. Panel, did you have any follow-up on any of the thoughts that were shared there?
Yeah, there is a literature article. The first author is Richard Chang, and that is focusing specifically on common deficiencies for topical products. So many of the things that were discussed in my talk and that Maria also mentioned. If you want more details, see that article.
All right, microphone number 1, nice and close for me, please.
This is Leandro from FDA, ODD, ORS, TQMM. I just want to roll back to the previous question regarding using PBPK to support non-Q1, Q2 packages. I think the presenters give very comprehensive answer. So my addition is that, we do for it's more case by case based assessment. Also, it's risk-based assessment. If the formulation is relatively easy, FDA here is not trying to make us a barrier to assess, to facilitate the process. And for the PBPK model establishment, verification is the key. And also it's risk-based for simple formulations. And you think there's sufficient scientific justification to support the case, we have a mechanism of pre-ANDA meetings. You are more than welcome to formulate a package and submit to the meeting request, and we are more than happy to discuss the case.
All right, thank you for sharing. Let's take an online question, please.
So this question might be for Dr. Burridge. Can I measure both API particle size and globule size using the same sample?
Yes, you can. What you'll need to do there, for example, if you're using microscopy, is find a way to distinguish the two particles. So for example, you might use bright field to look at globule size, and then you may need to use a polarizing filter to look at API particle size.
Okay, microphone number 2, nice and close for me, please.
This is for Dr. Rani. We have heard about QBD. Today, you alluded to bioequivalence by design. Was that a fleeting semantic, or is there a vetted thought process at FDA in formalizing it?
So it's a good question. No, the bioequivalence by design is not a formal program within the agency. It's actually a phrase that Charlie Diliberti coined, and I thought it captured well the intent of what we're trying to ultimately achieve with developing generic products that are Q1, Q2, Q3 the same, that you're building in the bioequivalence. So no, it's not a it's some kind of new catch program within the agency.
Okay, and our time is up, but before we wrap up, based on any of the thoughts shared, any final thoughts that the panel would like to share? Bianca, you got your hand there. All right. Okay, please help me thank our panel for coming and sharing.